Welcome and thank you for joining the first of 7-part webinar series titled Modern Particle Characterization Technique. This is the introduction webinar where we're going to give you a broad but informative overview of number of techniques. So today is introduction. Next will be the laser diffraction techniques, followed by DLS, multi-laser NTA, VET, zeta potential, and image analysis. So it is my great pleasure today to introduce you the Vice President of HORIBA Scientific, Dr. Mike Pohl. Mike received his PhD in inorganic chemistry and has dedicated over 34 years of his career in particle characterization. He runs meetings for ASTM and participates in multiple ISO committees. Mike is one of the hardest worker and brilliant thought leaders in the scientific community. His entire focus is set on solving customer problems. And outside of work, Mike is a grandpa living in sunny Southern California, enjoying life. Okay, Mike, I'm going to go ahead and pass the ball to you. Would you please share your screen?
Well, what we'd like to do today, I think, as Julie has introduced, is we're not going to talk about any of the specific techniques that she mentioned earlier, but what we're going to look at is how we use this data, how we generate good data, how we analyze what is happening with our particles. So we're going to use that kind of as the framework for what we're going to talk about today, and hopefully take you through some topics that are of interest to get you ready to go with the particle sizing. So here we go. This is going to be our topic for the day. Just to start it off, I think here we've given you a few examples of where it is that particle characterization as a general subject is important today. As you look here, I think you can see a whole variety of industries, all the way from basic chemical kinds of applications, pharmaceuticals, drugs, paper. So the first thing you kind of take away is the fact that virtually any industry that you have any familiarity with is going to be using particle characterization in some way, shape, or form. And so we'll show you a few actual examples here today and how we're going to use that data. So we'll get right into it right from the start. And here we're trying to illustrate to you exactly what is it that you're going to be doing with the data that you generate. We'll talk about generating the data a little bit later in our presentation today, but to start out, we just want to show you what to do with the data. And what we've plotted here along the y-axis is a physical property. And what that is, that's what you really want to know about your particles. And we'll talk about some concrete things there in just a moment. If you go down to your x-axis, you can see there's a lot of different ways to express your particle size information. You can be giving a D50, you can be giving percentage greater than 1 micron, all sorts of things. And again, we'll talk about these parameters toward the end of the presentation as to which one of these you really want to use or you want to pick for your application. But the key thing is then, as you're going through, what you want to do is you want to look at these particle sizing parameters and have these become predictors of the properties that are on your y-axis there, the physical properties. So as an example, we have their rate of surface removal. If you're working with CMP slurries, you're going to want to know when to stop doing your CMP process. And so in order to predict that, you're going to have to know the rate of surface removal. And so again, what you're going to do is you're going to have to look at your particle size parameter to give you a prediction of this rate of surface removal. So for example, if we use, say, the percentage greater than 1 micron for a CMP slurry, which would be the material which actually does the most abrading of all the particles in your slurry, what you're going to do is you're going to prepare samples with varying amounts of CMP slurry that contains material over 1 micron. And so what you're going to do is you're then going to go through, you're going to make up these solutions. Typically, you're going to spike them by having some separated 1 micron particles. and you're going to add those into the slurry, and you're going to vary those from, let's say, 1% maybe to 10%. And then in the background, you're going to take each one of those slurries and you're going to go ahead and you're going to make a measurement of your rate of surface removal. So those are, some totally different measurements that if you're in the semiconductor industry, you would know how to do. And so what you do is you prepare here half a dozen or so samples with different particle size properties. You look at the rate of the surface removal, and you then plot one against the other. And as you can imagine, this is quite a bit of work because you've got to make up these different slurries. You got to make sure you got the right percentages in there. And then you've got to go over and do the actual physical test, the rate of surface removal. But then you can see when you're done, you can just plot it up like you see here on the screen. And the whole benefit you've derived here is when you want to measure your rate of surface removal or know what that property is, you don't need to go through the whole process of doing that. That's a process that will take you hours to do, cause you downtime and so forth. Instead, what you do is you simply make one measurement of your particle size parameter, look at the percentage over 1 micron, go over to the y-axis, and you then predict what is your rate of surface removal. And so this is always going to be the case with this kind of a parameter. To just say I have 1% over 1 micron really doesn't tell you much about your sample. It gives you some rough idea of its physical properties, but it doesn't tell you how this is going to perform for your process. And so doing this kind of a correlation study is always where you want to get to, because at that point you just set a spec according to particle size, and at the end of the day you know what the rate of surface removal would be. But if you go again back to that y-axis, you can see there's many other properties there. You could look at the rate of dissolution of a drug, for example. You could look at protein aggregation to study that. You could look at pigments and study the hiding power of those pigments. In other words, how well they're going to cover your wall when you use them to paint with. So again, this is always kind of a shortcut to get you to that physical property that makes it easier, faster, all the normal parameters you want to see. And so this is where you always want to go. You want to spend typically weeks worth of time to get this correlation established, because once you have it, then you're going to save yourself a lot of time when you go to characterize your new particles that you purchase or you receive in a bag or however you get them. You're going to then just run the particle size, and you're going to know this physical property automatically without ever testing it. So that's where we want to go with this, and we're going to show you how to get the good data that's going to give you this correlation. So we're talking now originally about particle characterization methods. Julie alluded to a few of them. Here you can see them listed here again. Just a whole variety of different technologies. Some we'll talk about a little bit today, some we won't really have time to look at. But you can see what is critical is you really got to know what those particles look like, what are their properties, so that you can figure out how they're going to perform in your process. So what we're going to do now is we're going to switch over. We're going to drill down a little bit further into the particle sizing area. So here you can see what we put together is a list of the different techniques that are out there. Again, as you can see at the bottom, it again says many more. We just kind of highlighted some of the ones that seem to be most popular today. We'll give you a little bit of background in some of these. But again, this comes back to the original question that we were trying to address, which is how am I going to select the best technique or the best instrument to do my measurement? And so I think when we show you what is critical to each one of these measurements, in other words, what physical property does each technique measure, I think you're going to see after a while it'll become fairly obvious what's going to be the best for your particular problem that you're confronted with in your process or in your laboratory. So what we'll do is now we'll just jump in to show you these different techniques. Up at the very top, you can see we've got the different sizes that you may confront with your particles that you have in your laboratory. You can see on the right-hand side are the coarsest ones that you may encounter To the left-hand side are the smaller ones, which will be the ones that typically are of most interest to people nowadays. You can see the different applications here, all the way from powders that you would get in a jar, all the way down to macromolecules that you might have in a liquid system. Down at the bottom of the slide, you can see that you have a variety of techniques there, all the way from the more traditional ones, the electron microscopy, sieves, things like that, all the way down to some of the newer ones, the nanoparticle tracking, DLS, things along that line. So now let's start off into the techniques to show you what they have. Here you can see the Coulter principle. On the right-hand side, you can see you've got a small orifice there. You're going to be passing these particles through the orifice, and what will happen is it will disrupt the current that is flowing there, and the instrument is set up to maintain itself at a constant current, which means that the voltage will have to change to keep that current constant. And so as these particles pass through the orifice, hopefully one at a time, you're going to be able to get an individual signal from it, the amount of voltage that was needed to keep that constant current, and that's going to be proportional to the size of your particles. So again, you're going to have this in a conductive liquid. You're going to need to have this well dispersed because you don't want these particles sticking together as they go through the orifice. So each of the techniques we'll talk about, has certain pluses and minuses. There is no perfect technique out there that can handle every single type of sample. Here you can see we mentioned the fact that this originally came from the medical industry. This was used for blood cell counting. Not real big or very small particles, 7 or 8 microns, something along that size range. And so, very popular if you have narrow size ranges of particles. You don't want to plug up your orifice. If the particles get too small, you can't detect their signal. So as with every technique, a little bit of a challenge there. The next one we'll look at is Stokes' law sedimentation. Here you can see on the right-hand side, the whole idea behind it is, you're going to start out with particles suspended in a fluid. If you have different sizes, the different size particles will sediment at a different rate. You can see here the blue ones are the largest particles. They're settling relatively quickly. The green ones are kind of your medium-sized particles. They're taking a little while to settle. And then finally, your very small particles, the red ones, are going to settle the slowest. So again, here's your Stokes' law equation. You can see certain parameters you need to know. You need to know the viscosity of your fluid, the density of your continuous phase, your liquid typically, and then also the density of the particles that you're looking at in your analysis. So again, applicable to a lot of things. I think you can imagine if you did have samples that had somewhat mixed densities to them, it would be a little bit hard to segregate out different density materials because they would all be settling together, and so your data would get a little bit mixed up. So, some limitations, but again, if you're trying to do things typically over about a micron, the sedimentation times are reasonable. You can get a typically good analysis from this technique. Now, the one that we'll talk about in a later webinar that we're going to do is going to be laser light scattering. And here you can see the principle behind it is that little white box in the right-hand side there is actually your cell. In other words, that's where your sample is flowing. Typically, it could be either in a gas stream or it could also be in a liquid stream. And you're going to shine lights from different angles. And what you're going to do is measure the diffraction. So you're going to use different light sources. You're then going to measure at different angles. And after you've collected all the theory, and you're then going to calculate what your particle size distribution looks like. These measurements are virtually made instantaneously. You're typically measuring thousands of times per second. And so you can imagine you collect lots of data. You hopefully would get repeatable data from all the measurements that you made. You can measure a broad range of sizes here. You can go down well into the submicron region and go up to thousands of microns. Here's just one example on your page here. This is a very small silica. I believe this may be the one that comes from CMP. I'm not sure. But at any rate, you can see there's your distribution that you get. And again, most of these measurements are done in a minute or less. So, a very popular technique, very widely used. Now here we can see what I guess typically would be known as kind of the gold standard for your particle sizing, and that's going to be microscopy. I think as you can kind of see on the left there, it's not very big, unfortunately, but I think you can see you're actually looking at the images of your particles. So you can actually sit there, measure different dimensions of the particles. You can go through and count the particles as they're going in front of the camera so that you would have some idea of their concentration. You can cover, a broad range of sizes. Typically, your standard light microscope might be half a micron and up. And you can go down to things like SEM, where now you're going well down into the nanometer range in terms of the sizes. And so, again, if you measure some data and you want to say, geez, I just got 10 microns, does that seem reasonable? Certainly what you would do is resort to some kind of microscopic image of your particles. So again, it's very popular. You can do two ways. You can do it here. You can see with a microscope slide. That would be called static image analysis. You can also do this in a flowing system so that you would have an opportunity to flow the particles in front of your camera, look at them as they're falling in front of the camera, capture their images, and end up with something that looks very similar to what you would have from a microscope slide. Now, one of the things that has been around for a while now is you can actually go on the right-hand side of the screen, and there you can see we're now doing online digital image analysis. So all that you're doing is you're going on the left-hand side where you have a microscope slide, and in its place you have some sort of a flowing system of particles that will be measured by this probe. And so again, a lot of people, you'll find out later on, are going to have some challenges doing sampling, things along that line. And so what you want to do is you just as soon measure these right in your process itself. You know, obviously at some point you're going to put these in a drum or a bag or some rail car or something like that. And the big thing is, once you get it into the container, it's kind of a little late to decide you want to size those particles. So a lot of people would like to capture this information right during the process so they get a relatively accurate representation of what is with their particles. And again, we have a little section here a little bit later on. We'll talk about some of these different ways of doing the sampling that is going to become so critical. Now, another way you can do this, if things get relatively small, eventually what you're going to find out is that the particles become so small that a lot of the technologies are not capable of, actually doing the measurement. And so another way of looking at these very small particles is with dynamic light scattering. Here, you're now not going to measure the angular scatter of the light from the particles. You're actually going to measure the light intensity or frequency. And so here you can see this is some nano gold down at 50 nanometers. Here's a couple of runs that you have that gives you some very good repeatable data. So again, if you're looking for small materials, that might be a way to go. Now, as we mentioned earlier, a lot of other techniques we don't even really have time to talk about today. Today, we're just introducing you to the general subject, but I think just to illustrate to you some of the complexities of what you're going to deal with is on this next slide, the sedimentation analyses. These are all different ways that you can use to measure your particle size. So this is all the same technique, all Stokes' law, no difference there, but how do you make the measurement? If you go down the homogeneous technique, you just totally disperse your particles in the liquid and you measure them totally dispersed. With the line start, you're going to introduce all of your particles at the top of your measuring apparatus, cuvette, cell, whatever you have, and you're going to watch those particles all settle, separate themselves, and then measure them later. In terms of looking at your particle concentrations, now you have two different ways of doing it. You can use an X-ray, which is very good for inorganic type materials. You can use a light source, which is much better for doing organics, low atomic weight materials, which do not obscurate the X-rays very well. When you do the technique, two different ways of doing this. You could have your cell move up and down in front of, let's say, your X-ray beam to speed up your measurements so you didn't have to wait so long for it to settle. Or you could also have the X-ray beam itself just scan up and down throughout your cell so that you could, again, speed up the analysis. What you're going to find is you get down toward, let's say, 1 to 10 microns in terms of size. If the particle doesn't have a very high density, it could take you sometimes as much as hours to get the sedimentation to happen. So, either moving the cell or the source are designed to save you some time there. Then again, another kind of time-saving way to do it is here, the next two. We talked about basically gravity sedimentation, where you just watch it settle over time, but you can also speed this up with a centrifuge. So instead of having essentially 1 g of gravity causing your particles to settle, or I keep saying settling, I should really include rising in there. In other words, if you have some of a density of let's say some droplets that are less than 1 and they're in water, those particles are going to rise to the top of the cell. They're not going to settle. But in either case, you can do that, for example, with a centrifuge. So in essence, what you've done is you've taken 1 g of gravity, which is what you have available to you in your laboratory, and you've expanded that into as many as thousands of g's if, again, you have small, low-density particles. So again, I don't want to dwell on this too long here today, but I just want to give you some idea that this is a pretty complicated field. It takes you some time to understand all the things that are being done, and I want particle sizing, I want sedimentation. Okay, now how do I want to do it? Gravity or centrifugal, light or X-ray, so forth. So just again, giving you a little bit of a flavor for what you're going to be confronted with when you actually go ahead to do, your analysis of your actual particles. Now, the one other topic we're going to talk a little bit, this is now no longer particle size, but is particle characterization. But here you can see the BET surface area. And I think what you're going to find is very, very often this technology goes hand in hand with your particle characterization, because what you're looking at here is sort of the hand-in-hand partner with this particle size measurement. Because what you're going to find happening, and we'll talk about this here in this PowerPoint in just a moment, is that as your particle size gets smaller and smaller, your surface area is going to end up getting larger and larger. So these are two sort of complementary techniques that can allow you to measure, in many cases, the very small particles. And so here you can see in the upper left-hand corner, that's a visualization of what is happening at the surface of your particle. Those little blue light-colored dots are actually representing molecules of nitrogen, and those molecules at very, very low temperature, at liquid nitrogen temperature, are going to adhere to your surface. And so when you want to get your surface area, you could typically sit there and wait till every single little area of the surface was covered, or in many cases, you only go to a certain stopping point, and then you sort of extrapolate how much more nitrogen would it take to totally cover this surface. And so that's a representation of what's happening in your tube as you're making your measurement. At the bottom, you can see what's going to be happening. If you have the first peak that you see is a peak that comes in from the air that's present in your sample. But then the next peak, as you can see here, this is a small surface area material. What we're basically looking at is we're looking at the area under the curve. And initially, what you do is you're going to take your sample, you're going to degas it to get everything off the surface so you have a clean surface. You're then going to dip that into a liquid nitrogen bath that lowers your temperature down, and now the nitrogen will begin to absorb on the surface. And so what you can see is that area under that peak is the amount of nitrogen that got absorbed onto the surface. Over to the right a little bit further, you're eventually going to take your liquid nitrogen dewar away from the sample. When you take it away from the sample, now all the nitrogen gas that was absorbed on the surface is going to be desorbed. So you have an absorption peak on the left, you have a desorption peak on the right. Now, the next graph over is your large surface area materials. And what you can see here is that obviously the heights and the area of each one of those two peaks is quite dramatically larger than a small surface area material. What you'll also notice is one of the parameters that you typically want to look at is typically you either want to measure adsorption or desorption. You don't really need to have two values unless you have some special work you're doing. But what you can see is on the left-hand side there, on the red curve, the first thing that happens is, again, that's your adsorption. And what you see happening here is that it takes a long time to get to the very end of the adsorption curve. And so what you're confronted with is if you want accurate, repeatable data, you want to have the area under that curve as condensed as possible to give you a much better ability to integrate what is there. And so when we go to the right side and we see the desorption, you can see a much more narrow, much more concentrated peak. And so in most cases, that would be the peak you would want to look at. You look at that desorption peak because it happens so much more quickly. You have a much more area under the peak happening very quickly, so it's much easier to integrate. And there you can see on the right-hand side what happens. Typically, if you want to do a multi-point surface area measurement, you're going to use different relative pressures of your nitrogen to make the measurement. And you just go through and do these sequentially in order to calculate your surface area. So again, we're not going to spend a whole lot of time on this. I just wanted to introduce it to you because it is one of the subjects that comes up in one of our later webinars. Now, one of the things I wanted to kind of introduce to you here is just what is going to be involved in your getting your sample ready to be analyzed and to report the data. You can see we've taken this from a publication that came from NIST, and this gives you a very, very good idea of the challenges. In other words, somewhere out in a process, in a mill, you've got some great big bulk sample. You know, when we look at the analyses we're going to do, they're typically only going to require grams of sample. And so you could be confronted with potentially as much as thousands of pounds of material, and somehow or another, you've got to come up with this representative sample to put into your instrument. So the first thing we'll talk about is going to be this subsample preparation. How do I get a subsample? What do I do to make sure that it's, you know, representative, things like that. Next step is going to be we got to get this powder ready to measure, whether it's wet or dry. One of the challenges we'll talk about is you really got to determine, do I want to see this particle as it sits here in my beaker, or do I want to disperse it and look at the fundamental particles which are really in this sample? So in either case, wet or dry, you're going to have to go through some sort of a dispersion protocol. Then you're going to go through a sample preparation step. Again, we'll talk a little bit about that in a moment. You're going to want to make sure, you know, that your instrument is working properly. So you're going to be calibrating it. Now, not necessarily before every sample, but in some regular basis, you're going to want to run a calibration. I would call it almost more of a check sample so that you know that everything's copacetic. Nothing from yesterday's analysis got stuck in the tubing or gummed up your cell or did anything crazy in there. Once you've got that all completed and, you know, verified, now you're ready to conduct your measurement. In most cases, again, you're not going to just accept one sample. You're going to want to do proper statistics to get the best possible data. You're going to collect the data. You're going to do some processing of that data. Then you're going to get to the point where you have to interpret the data. Again, we'll spend a little time on that. And then last but not least, reporting your results. So this is kind of a flow chart of what happens and kind of lays out what's going to happen for you in the rest of the webinar today. So up top is something that, is 100% under your control. You have to get that sample ready to go, get it prepared before you put it into the analyzer. Now, as you're going through, number one is certainly you're going to have to be sure that you do have that representative sample. And what you'll see as we go through this is it seems like it should be relatively easy to do this, but in reality, it's really quite complicated because most particulate systems are not homogeneous to start with. And so the challenge really is to start with a non-homogeneous material and then end up with something homogeneous as it's going into your analyzer. You'll see from the subsequent slides that, this is almost a science in and of itself. There's textbooks written on it. There's different ways of doing things. Bear in mind, this is going to be very, very important, especially if you have a broad distribution of particle sizes. The broad sizes very, very definitely segregate, and temptation would certainly be, here's my beaker, let's just scoop it off the top. But as you'll find out, that can be very, very dangerous. And, when we look at people who are having problems with particle size, very, very often this is the place where they got tripped up. So here we've got an example. This is a mixture of different sizes of sand materials. Again, I'm not going to go through all of these individual techniques. You can see them. They're listed on the left-hand side here. This is some information we lifted from Dr. Allen's book. But I think the key thing you can see here is that, your sampling determines your standard deviation, basically. I mean, there's always going to be certain random variation. You can see at the bottom there. You're probably never going to get down that low. But again, typically you want this to be as tight as you can. And so the spinning riffler is going to be the technique that will get you there. So one of the things that, you're very, very often confronted with is you have a bag or a drum full of particles and somebody says, what is the size of this sample? Now, what we're illustrating here is what to do when you're handed that drum or that bag or that beaker full of powder. But the whole point of it is this would not really be the way you would be recommended to go. In other words, at some point in the process, you were actually doing the filling of the drum, the filling of the bag, things like this. That would really be the point where you ought to do a proper sampling. And what you want to do is, as you're filling this drum or bag or rail car, whatever it might be, is every so often you want to take a small fraction of what's going into the container. And you do this until the entire vessel is full. You then combine all of those individual samples, and that is the composite of what's in that entire container. But the real world is very often you don't have the option to go back to the process, grab the particles, do things with it. So this is the next best way to do it. And I think you can see even the techniques here are very related to the best technique, which is pulling the small samples over long periods of time, is what you're going to do. You can see these drum thieves are being put into the particles. And what you're going to do is you're going to go to different areas of the drum. You're going to go to different heights in the drum, let's say. You're going to open up your sampler. Particles are going to run in. You're going to close the sampler, pull it out, put it in a separate beaker, a new beaker. Do this a number of times, and this way you should then end up with a representative sample of what was in that drum. So again, something that people very often don't think about. You open up the lid of the drum and you scoop something out and you say, here's my sample. That can be very, very dangerous. So here's another example of a chute riffling. And here you're just going to allow the particles to go down. You can see if you look over on the right, there's 4 different slots that the particles are going to come out in. You split them up, again, hoping that, some of the large, medium, and small particles go into each one of those slots. And so we saw earlier, this is certainly better than just scooping or coning and quartering, but certainly does, leave a little something to be desired. Here's some pictures of some so you can see. The one on the left is the one we really want to talk about most because this is really your spinning riffler. In other words, up top, if you look there, it's a little bit hard to make out, but at any rate, that's a funnel. The funnel is feeding your particles at a certain rate of speed onto a vibrating tray there. Those bottles, which are down a little bit to the left of the chute, those are spinning around at a certain number of RPMs. And so you capture a little bit of the sample, goes into bottle one, a little bit into two, three, four, back to one, two, three, four, back to one. So what you hope is that if what you dumped in there, which is not homogeneous, which is most of the cases, you will get a little bit of each of the layers of the particles into each one of those bottles for further analysis. So again, lots of different ways to do this, lots of different types of instruments out there. But certainly if you want to get repeatable data, you certainly got to be paying attention to this, or you're just going to get what was on the scoop rather than something that really represents what was in the entire drum. Even, you would think, gosh, a liquid stirring in a beaker should really be something easy to sample. But again, you have the same issue. Things close to the impeller are going to be different than what's up at the top of the beaker, what's at the bottom of the beaker. And so again, you are going to have to go to different places in the beaker to get a good sampling, mix those materials together, come up with a composite sample that you can now hopefully stick directly in your instrument without having to touch it further. Some cases you may have to do this one or two or three times. If you have a very, very concentrated slurry and you're trying to dilute it down to 1/10 of a weight percent or something like that, that you would use for laser diffraction. Now this looks really complicated. I don't want to try to take you through the whole thing. Let me just give you the highlights. What we're starting with in the upper left-hand corner is 3 containers, say drums of particles. And what we're trying to do is we're trying to make a particle size reference standard, and that's all the way down in the lower right-hand corner. And so what you're going to do is you're going to have pounds or tens of pounds, whatever, coming from those drums. And now you want much smaller bottles on the lower right-hand side. Particle sizing standards don't last forever, so you don't want to be putting 5 or 10 pounds in a container because it'll go bad before you get to use it. So these are made up in much smaller sizes. And the point of what we're trying to show you here is that what you've got to do is keep cross-mixing these many times to get to the point of having a homogeneous sample. And so you can see we started out calling, 35 kilograms green, 35 blue. Okay, they went into individual containers. Then each of those individual containers had to be mixed together so that you had some from the blue drum and some from the green drum all getting mixed together. Then you're going to take those, you're going to have to divide those up and now make those into 2 different lots, an orange lot and a yellow lot, and so on down through this whole schematic. And certainly the average customer is not doing this. This is, you're talking about weeks and weeks of work, but it just shows you that if you want to get very, very repeatable types of samples, you have to go through a lot of mixing and analyzing and so forth to get good data. So again, just an illustrative example, but shows you what steps you have to go through. Okay, now that we've got the sample representative, we're ready to go. Now what's next? Well, sample preparation. The most typical thing you want to do is you want to prepare the sample, how it's going to be used after you do something with the particles. In other words, those particles that you're analyzing are going to go into a process, are going to be used in some other fashion. And so if all you do is dump it in and use it exactly as it is, you don't want to use energy to disperse it. If in point of fact you're making paint and it's going to get some very high shear environment to get it to mix up properly, you're going to need to use some energy for your TiO2 particles. Some other things you can consider: what are other people in your industry doing? We'll show you, we have an example of that a little bit later on. Who's going to look at the data and what are they going to be interested in seeing? When you look at the data, we'll show you some here in a minute, there's a lot of information there. How do you kind of condense that down to be in a usable format? We mentioned this primary particles, or how they truly are. You're going to showed you a little bit, just like with sampling, there's quite a variety of tools at your disposal to disperse the material. And then ultimately, what are you going to do with the data? You're going to match the data from another instrument, another technique. You're going to be worried about the performance. You want to get the performance prediction. You're doing the research. So just a whole variety of things you have to think about as you're doing this, because all of these will affect how you're going to choose what to do with it. So here's a listing of some things. These are typically for wet type of samples. Variety of media you can use, could be organics, inorganics, whole variety of dispersants. Heck, this McCutcheon directory is a whole book full of different things that you could use to help you out to get your particles dispersed if you want the fundamental particles. You have all sorts of mixing devices you can use, magnetic stirrers, whirring blenders. You can go up to high energy. You can go with ultrasonic probes and baths and so forth. Some of the instruments you'll look at recognize that you might have unstable samples, and they actually will keep dispersing your sample as you're getting ready to do your analysis. So again, a whole variety of questions you're going to have to answer as you go into this. Here, this is actually a kind of a logic tree for doing dispersion, comes from ASTM committee B9, the powdered metal folks. Again, I don't want to go through all of this with you, but you can see you got to get your sample up top. Make sure that the sample's not reacting with the liquid medium you have. You have to make sure it's not liquid soluble. You have to pick surfactants, either organic case or the water case. You have to make sure everything is well wetted. And then at the end, once you're all done doing your dispersion, you got to analyze it to make sure it is perfectly stable. And so this gives you an idea, if you're confronted with a totally new sample you've never seen before, what sort of a reasoning or logic do I need to go through in order to get down to having a dispersed particle to measure? Now, as we mentioned earlier, if you go out in the literature, if you belong to ASTM or some other trade organization, what you're going to find is these people have figured this out already. So rather than you reinventing the wheel, you just go here. Now, again, these are examples from B9, powdered metals. Probably most of you are not doing these, but just use it as an example. If I'm looking at tungsten metal powder, I could do this in water, 0.01% sodium hexametaphosphate, probe it, ultrasonic bath, so on and so forth. And so again, you can save yourself a lot of time. You just look these things up and don't try to approach it. I mean, it could be a new research sample nobody's ever dispersed before. That certainly is within the realm of possibility. But again, a lot of cases, just go and look in the literature. Now, here's what we talked about earlier. This is what your data is going to look like. This is your cumulative distribution. And again, you can see, I mean, there's just tons of data here. You've got a curve. The most typical ones you'd want to look at is 0.1 or 10% of your particles. You could do the median, the D50. You could look at D90. Some people are worried about the very, very large ones. They might be at D95 or D99, something like that. Once you get out there on the edges of the distribution, it gets a little bit less repeatable and a little bit more questionable. But again, you're going to have to make the decision yourself as to what you want to look at. Am I worrying about a few scratching particles in my abrasive? Am I worrying about maybe too much oiliness to my emulsion that I'm preparing? You might be down at the D10. Is the distribution too broad or too narrow? Maybe your particles don't perform well when they're too narrow. So again, this is all going to be somewhat relative. This is, the reason that companies like HORIBA are around. When you talk to us or send us samples to analyze, we'll help you figure out where you need to be and what you need to look for. So again, this is probably a whole talk in and of itself, but I just wanted to kind of give you this as a highlight. So what we can see from what we've addressed is we could conclude whether the distribution has become larger or smaller. We can find out whether some part of the distribution has changed. Is it now bimodal? Does it have a small tail on it? What's going on? Are the distributions themselves broader or more narrow? If you do have two modes, you typically would want to look at those separately, not analyze them together because each one has its own statistics. You could look to see if you have too many coarse particles to sample, just look up at D95, something like that. You could worry about whether two sets are similar or different. You could compare some of the statistics and say, yes, this is the same as supplier's A material, or no, this is really different than the other supplier's material. I really don't want to use this material. And you could also assess the techniques. You can look at several different of these and see which one best helps you predict the performance of your particles. So again, we're coming back to where we started. Here's the series of size ranges across your x-axis here. Here's some of the different particle characterization techniques listed here. As you probably figured out, these are all ones that HORIBA offers, but it gives you some idea of just a broad range of sizes that can be handled, the different technologies that are available, and we're here to help you decide what would be best for your particular types of particles. So that covers about the subjects I wanted to talk about today, Julie.
Thank you so much for the excellent webinar. That was very informative. I want to let everyone know that this presentation is recorded and will be posted on our website, along with future and past webinar information. And then we'll move on to the Q&A session. All right, question. How is particle size related to slower excretion rate?
Well, I mean, sounds a little funny, but I mean, we've done virtually every kind of sample you can imagine. And for example, we've looked at, samples coming from animals to look at the size of the particles to determine how long this material spent in a digestive tract, things along that line. You can look at the rate of liquids moving through films, through porous materials. If you're thinking about, sweat, things like that, we've done things like studies with people riding bicycles and, looking at the rate at which the droplets are coming out of their skin, so forth. So, it's like everything else. You have to just do your correlation again, the property you want to know relative to the size.
Thank you. Okay. Question 2, laser diffraction technique. Would it be able to detect particle in 2 micron range in presence of larger particles, such as 100 micron?
That is the challenge, because if you look at laser diffraction, the particles that are large scatter a lot more light than the small particles. And so there can very often be a tendency to miss the small particles. You're so focused on the big ones that you don't even have enough little ones in there to make the measurement. So there are different ways you can adjust for that. One of the typical ones we would do is we would do two measurements. So we would do one measurement where we look at typically the whole broad distribution. Then another one, we would put in a more concentrated sample and we would totally ignore the large ones, only focus on the small ones, and in that way make sure that we actually saw those and we had some idea how many of them were actually present in the sample.
Thank you. Okay, next question. What role does PDI play in particle size analysis using DLS?
Um, that can play a large role in it. To be honest with you, it's not an area that we have a whole lot of experience working with. So typically, I mean, we would come back to our normal response would be send us some samples. Typically, we would like to see some good, some bad, and some intermediate samples. Let us show you if we can come up with some data that correlates with what you want to measure.
So Mike, I have Jeff in the background and he would like to make a comment regarding to
Okay.
this question. Go ahead, Jeff.
Yeah, so I'm going to take a quick guess that he's asking about polydispersity index, which is the breadth of the size distribution. And so when you do a dynamic light scattering measurement, you'll often extract a z-average size and a width of the size distribution. If your size distribution gets particularly wide, DLS starts becoming more difficult, and then you'll shift over to techniques like multi-laser nanoparticle tracking analysis. So I guess there's two comments. One is PDI's way of keeping score about how wide is your distribution, and second is that if that gets to be too difficult, then you'll start reaching out to other techniques for broad size distributions of nanoparticles, like multi-laser nanoparticle tracking analysis. Okay, thanks.
Thanks, Jeff.
Thanks, Jeff.
And if you're wondering, Jeff is our product line manager. He's also our speaker for the Particle Characterization series that takes you through multiple steps of considerations when looking at particles with laser diffraction. The next question is, what is the best method for dry metal powders?
You know, it's the old story. If they're free-flowing materials, dry would be a perfectly fine way to do it. If they become particularly sticky or agglomerated or things like that, typically going with wet would be a bit better because you have some more tricks up your sleeve to do the dispersion. So, you know, with dry, you can do a few things. You can use some pressure differences to get them to disperse. You can put some flow aids in there, but those typically are not quite as effective as what you have wet. So if you have something tough to disperse, I would typically recommend wet. If they're easy, free-flowing, dry should be the best way to go.
Thank you. And that kind of takes us to this next question. How frequently do you have to clean the instrument? And I'm assuming this is for laser diffraction.
Well, I guess there are probably two answers. One would be the wet and one would be the dry. With either one of them, you actually have a built-in way of analyzing whether you cleaned up properly or not. Because what you're going to start out with in both cases is you're going to start out with a measurement before you even put the sample in there. And so you're going to have a good idea exactly how much light is coming through the sample. You put your sample in there, and whatever value that comes to, let's say 80% T, what should happen is when you're done running the sample and you clean it out, if you put that same sample back in again, it should come right back up to 80% T. If in point of fact it ends up being 70% T, that tells you you have some leftover particles somewhere in the system that you have to get out before you run your second sample. If the number goes up, that's telling you you've diluted your sample. So somehow or another, as you were draining your fluid, you didn't do a particularly good job of draining the fluid, should drain and start all over. So the instrument itself kind of tells you when it needs to be cleaned just by how it's behaving.
Thank you. And then I think we have time for two more questions. I just want to insert this in first. You can also send us questions at labinfo@horiba.com. Okay, the next question is, what's more important, determine the number of particles or the volume distributions when it comes to dry particles?
Well, I'll tell you, that's something that in a lot of cases you end up really wanting to know both of them, because as you're using the materials, you know, you're going to want to use so many milliliters of such and such a concentration. And so in certain cases, the count would be important. In certain cases, you just want to know the distribution and you don't care about the total number of particles. So some techniques measure one, other techniques measure the other, and it's really going to depend upon what you're going to do with the data, which one would be more valuable to you.
Thank you. So going back to what you were talking about regarding to cleaning, is there an automated self-cleaning mode after the analysis?
Yeah, I mean, you can set it up to where it checks itself. For example, I mean, at the start of the day, you're going to do a rinse of the whole system to make sure everything gets well wetted. There's nothing stuck in there. You're going to run your first sample. You're going to drain it out. You're going to put some more of your clean fluid in there. And so you can automatically program it that when you put the second fluid addition in there, that you want it to be, say, between 99 and 101%. So you can program it to do that checking for you so you don't have to do it, or you can just glance at it after you've cleaned it up, put the fresh fluid in there. You can just look and say, yeah, this is 99%. Now, one thing you want to be careful of, to be honest with you, is that the instruments typically what they do is if you wait long enough, they're going to re-zero themselves. So if you put something in there that has 95% T, it's going to ultimately, if you don't do anything about that, it's going to re-record 95% T as 100% T. So there's certain caveats, but yes, it can be automated in the system.
Thank you so much, Mike. I think we have a few more questions, but in the interest of time, again, please feel free and don't hesitate to reach out to us and send your questions to labinfo@horiba.com. I want to encourage everyone to sign up for the newsletter so you know where to sign up for the next webinar on modern particle characterization techniques, on laser diffraction specifically. That's going to take place on May 28th, same time. Okay, before we end, Mike, do you have anything else that you would like to add?
No, I would just say that basically our job here is to help people to learn how to use our equipment. And so, our analytical laboratory here in Irvine, California is really here for our customers. We don't do any paid analyses. We don't do anything but support our customers, help them figure out solutions to their problem.
Absolutely. So take advantage of it. I think that's what I hear. All right. Thank you so much, Mike. Thank you, everyone. For now, have a great day, and we'll see you at one or more of our future webinars.